As we begin to discuss invertebrates, and vertebrates eventually, we're going to keep referring back to our phylogenetic tree. The reason is we want to guide you through this process based on derived characteristics. So, our first stop is sponges, or porifera. Why do we stop here first? Well, as you see, on the right side of our tree, we have tissues in symmetry, meaning that those are derived characters for the organisms on the right side of our tree. But if we actually start to the left, we don't see any of those derived characters, meaning that sponges or porifera, phyla porifera, have no tissues, no symmetry. So what do they have? Well, they have no organs, they have basic cell structures, and when we look at the sponges themselves, they're a fairly simple structure. However, they are very, very good at filtering water, as you might expect from something known as a sponge. And when we look at the damage to a sponge, I have here that they can actually reform. That's an important characteristic when we look at organism complexity later on. So we're simply looking at the ability of the sponge to bring in water through its sides and push water out through the top, and in the process what it's doing is filtering through these cells that are around the outside of the sponge. And when we look at moving this water through, we're not only looking at getting nutrients, of course, but also oxygen, and then eventually getting rid of the wastes as well. This central pore is known as an osculum, and depending on the sponge type, it can come in a variety of shapes and sizes, of course. Some sponges can be enormous, but when we look at these guys, they have the basic kind of cylindrical structure to them. So moving in that water brings in oxygen, brings in food, and helps the sponge get rid of waste. And so what we're looking for is that water flow through cells that will allow the surface cells to actually pick up the nutrients that they need. So these little flagella, or little fingers, are going to be acting as filters and picking up these particles as they move through. So in this, we do see the skeleton per se of a sponge. Remember, they don't have bones, so their skeleton's actually made up of something known as spicules or even collagen. So you know collagen from your own system gives it elasticity. In this case, we're looking at this as being a supportive structure for the sponge. They do go through sexual reproduction most of the time, but we can also see asexual reproduction as well. So when you're thinking about these phyla, you need to think about things like how do they eat, how do they get the nutrients they need, how do they get oxygen, how do they get rid of waste, but you also need to think about how do they reproduce and what is that basic life cycle associated with them. So sexual reproduction is as you might expect, simply releasing sperm and eggs into the ocean. Asexual, they simply bud. So a piece of them may break off and start a new sponge. So we end up with a lot of colonies of sponges, so kind of a field of sponges, so to speak, in the ocean. So this is a sponge actually going through sexual reproduction, so releasing eggs and sperm up into the ocean, and you see it's quite a cloud that's being released. This is actually done in cycle with the full moon, so many divers are interested in seeing these kinds of events and will go down particularly associated with full moon cycles and see some of these spectacular events. So the egg and sperm are released into the ocean, meaning that they have to find each other in the ocean, right? And when they do, they go into the blastula stage as we would expect. And they actually go into this kind of mobile larval stage, meaning that once they start to develop, they'll actually keep floating through the ocean until they find a space to settle on and have developed far enough to actually settle down and start to grow into a sponge.